I think we are ready to go, and we have 34 participants already joined us, so might as well get started, John. Up to you. Please introduce yourself and then go ahead with your
okay, thanks.
presentation.
Thanks for that. Look, I'll just start by sharing my screen to make sure that it works. Are you seeing a shared screen? Hello? I just want to make sure you can see my shared screen. I'm not hearing your audio right now.
I can see your shared screen.
Very good, thank you.
And I can also see your mouse moving, so that should be fine. Great.
Okay.
So now I go
All right.
mute.
So I'm John Rugis from University of Auckland. I work in a research group with James Sneed, also from U of A, as well as with David Ewell from University of Rochester in the USA. James and David have been working together for, well, as James describes it, since the beginning of time. So relatively speaking, I'm a recent addition to the group. My title, as you can read, is Processing Calcium Signaling Fluorescence Microscopy Image Stacks. So it's about cell biology. I'd like to start by highlighting the value of interdisciplinary collaboration. And in our case, my background's computer science. For James, it's mathematics, and for David, physiology. So in my joining the group, I've brought enhanced computation, well, for model building, simulation, visualization. And I'll say that the group's broad topic coverage was probably why the project was awarded the latest grant. I mean, you never really know why these things are accepted or rejected, but we speculate that that's the case. So the work is supported by a grant from NIH, so that's National Institutes of Health in the US. Just a little bit of biology background to help all this make sense. Calcium signaling occurs within the cells of the parotid gland, which is responsible for production of saliva. So calcium signaling helps facilitate lots of the activity that occurs within the cells, including kind of creating sort of like little water pump cells. It's kind of how you get saliva. David's labs recently acquired in vivo microscopy capabilities. What that means is it's pretty amazing. It can actually image fully intact live cells in a functional gland in a live animal. It's amazing, and acquire imagery in real time. And the extracted information from the microscopy work is used both for direct analysis and in building mathematical models for simulation. But in this presentation, I'm just going to focus on work done with microscopy image stack processing. So, okay, here's what an example image stack looks like. Each slice or each image in this stack is a 5 by 12 by 512 pixel image. It's just sort of a 2D cut through the specimen. And with the latest hardware, these are acquired at up to 20 images a second. A typical experiment will run up to 30 seconds. And each full experiment typically produces 4 image stacks because they stimulate the animal with different electrical frequencies to induce it to produce saliva. I'll play a little movie of this image stack. It's playing back in real time. About 10 seconds into the video, you'll see flashes of light, shall we say. They're regions of cells that are fluorescing due to an increased concentration of calcium. Near the end of the video, the calcium dies away because that means the stimulation stopped. I'm going to back up to about there in the simulation and say that the small dark disks that you see are cell nuclei. Cells are organized into little clusters. They're called acini. It's kind of like the shape of a potato. Hopefully you're seeing my little mouse pointer hover around those. And the calcium lights up in regions that are along the surface of cells and kind of like form fingers around each cell, sort of like they're grabbing each cell. And the region of interest, or regions of interest that we're after in these images, is just each of those little fingers separately. So you see that in this image stack, there's dozens of these little fingers. Anyway, image stacks from a single experiment easily consume over 1 gig of storage. And in the past, analysis of data from a single experiment took up to several weeks. You can imagine the difficulty in extracting or even just identifying fingers and extracting information. Anyway, the goal from a computer science point of view was design a custom computational workflow to, in the first instance, semi-automate the region of interest determination, and then to do subsequent data processing and analysis. So what I chose to do is, as you do with any given task, is a tool set, and I chose to use a collection of Jupyter Lab notebooks, Python for computation, Matplotlib for visualization, and to leverage extensive Python scientific code libraries. Rather than shovel all this work into one notebook, I'll split them into 3 categories: pre-processing, analysis, post-processing. So pre-processing, image stabilization; analysis, image noise reduction. You would have seen on that first little video I showed that imagery is quite grainy. Automatic region of interest determination, calcium plots for regions of interest, summary plots, and some post-processing as well. Let's jump into a quick look at, well, they're just screenshots of notebooks rather than live demo. The way I've organized this is whenever you see a black rectangular outline, that's a Jupyter Notebook screenshot. Some kind of cool things about Jupyter Notebooks, if you're not familiar with them. There's code hiding. In other words, the code that produces everything happening in this notebook is embedded in the notebook, but you can hide the code, and you can build context-sensitive GUI user interfaces. So in this case, because the notebooks are destined for a lab environment, the paradigm of lab notebook is a really good philosophical fit. The user doesn't have to type in anything. Again, there's context-sensitive GUI input for things like that. Here's one of the code blocks opened up so you can see what's in it, so that at any time the code becomes visible. So for things like, you might say, overall transparency in research, with notebooks, your code's just automatically there. It's embedded in the notebook itself. Definitely organize things such that the user of the notebook doesn't have to alter the code to adjust any parameters. They're just done in a GUI that's also embedded into the notebook. So the development of the notebook happens quite closely with the users in David's lab group. Results are organized, or you might say mandated by the use of the notebook, in that whenever you run the notebook, the analysis notebook, it creates timestamp results directory. So the user doesn't have the same burden of data management that would otherwise occur without using the notebook. Okay, into some of the analysis. For this region of interest determination, the first thing I did was determine essentially the static image. What does that look like before the cells are stimulated? Because in fact, in some sense, that could just be viewed as a base ground truth that from the point of view of stimulation, we don't really care about. For diagnostic purposes, I've included pixel histograms for all these images. So we have unstimulated, then an average image just during the stimulated time period. So now you see the regions where it's red, where stimulation highlights occurring. But the real magic is this kind of time difference image, where now just in red, we've got the stimulated regions, and that's what we're after. That was used to create what in computer graphics are just known as masks. So they're just sort of like in the mask, wherever you see white, those are where a region is, and those are the pixels that we want to further analyze. The code applies some filtering that to clean things up, uses some nice morphological operations to isolate out the fingers, and then, among other things, tells you how many pixels go with each region. So for example, region 1 has 138 pixels in it, and so on. Again, the code is embedded in the notebook. So yep, to do this, I've used binary erosion, binary dilation, and so on. Given regions, we can use those region determinations to extract pixels out of the image stack. And just for 2 of the 18 regions, we're now looking at plots. They're actually a fairly surprisingly diagnostic tool, because for region 1 in particular, we see that with all the 255 pixels plotted in color, some of the pixels were clipping, and those were used to refine the lab process. But anyway, the real interesting research info is when you average all those pixels together, you get some very nice oscillations, which are what was expected. What the researchers were really after was summary data over a number of different image stacks associated with different stimulation frequencies, and we were able to provide those nicely with this one particular notebook. Additionally, we thought, well, let's further analyze these results. There's oscillations there, but how can we further characterize the nature of those oscillations? A fairly obvious choice is to do frequency analysis of those sections in the plots, which we did. And in the first case, we applied FFT analysis, so that's fast Fourier transform, but some nice results out of that, although I'll say that it wasn't as clear or simple to interpret as we might have hoped. However, even with that said, one problem with an FFT is the kind of discrete nature of the results, particularly at lower frequencies. So an easy way of saying that is these plots are kind of choppy. We came up with another approach to frequency analysis that uses filter banks. Gives much smoother results, but still not particularly easy to interpret.
Jen, you got 1 minute to wrap it up, please.
Yep, thanks. So with that said, we cooked up another way of characterizing the results known as peak counting, which I'll quickly say worked out very well. And the code for that is in the notebook. So it's reproducible and transparent. One further thing we did was produce videos, summary videos, and rather than play through the whole thing, I'll zoom through part of it and say that what you see is while an image stack is progressing, you can see a plot progressing as the video is showing. So it's kind of a nice thing to show at conferences, as I'm doing right now. In summary, overall, problem was solved. Lab notebooks worked out very well. Initial analysis can be done in under 20 minutes as opposed to a couple weeks. And file organizational structures now reduce data handling complexity. Slight plug for our research in open science. All our notebooks are hosted under GitHub. Results are reproducible, transparent. This openness is encouraged by our funding agency, and it's personally rewarding to share. In summary, we've combined physiology lab, math modeling, computation, and visualization within our group. Special thanks, Center for E-Research, U of A, and NZRSE for compute infrastructure and consultancy services. Thanks for listening, and sorry for going over a minute or two.
Thank you, John. Just a quick question from Cameron Kerr. Does your workflow integrate with any image repository? And if so, does your data capture protocols use Jupyter, or is that a different process?
Okay, so right now, well, the first thing I'll say is that in terms of imagery, the primary target for imagery is to go in publications. We would secondarily then make the images in publication available in a repository. And then what was the second part of that question again?
Does your data capture protocols use Jupyter, or is that a different process? In other words, your data capture.
Yeah, the data capture is really driven by the equipment in the physiology lab. So they use Olympus microscopes, and there's software that comes with those that writes those out in a proprietary format, and that's that. I mean, the research group has no control over that. So part of our workflow converts Olympus format to a very generic TIFF format. And we use a secondary tool for that. We use Fiji, which is ImageJ, a well-known image processing, microscopy image processing tool set.
That's fantastic. Thanks a million, John.
You're welcome.
Thank you. Mingrui, over to you.
Oh.
Cool. Thank you very much. And let me share my screen first. Right. Can everyone see my screen okay? Cool, thank you. And good afternoon, everyone. My name is Mingrui, and I'm a PhD student in physics from Massey University, working with my supervisors, Elke and Joachim. Today, I'll be talking about the programming language Julia and how to write a parallel code with Julia. I will also briefly talk about the Monte Carlo algorithm I use in my daily research for physics. And lastly, I will tell you a bit about a profiling of a parallel Julia code on NZRSE. About Julia, Julia is a high-level programming language. Simply put, it is very new and it is pretty fast. And it has a great number of packages already available, covering a great range of categories. And we have a very active community, and the community is rapidly growing. Also, it is free under the open source license. So the most interesting feature for me is the design of Julia to write a parallel computing code. About parallel computing with Julia, there are like many options you can actually choose from. The first one we can see here is called the distributed computing and with the built-in Julia package called distributed.gl. Also, you can use a multi-threading feature that's available with Julia version since 1.3. It's also a built-in feature. Alternatively, you can use the MPI with MPI.gl. That's a wrapper of the external C MPI library. In today's talk, I will be focusing on only the distributed and MPI because they are using distributed memory, and I want them to work across multiple computer nodes. As we can see here, the structures of these two parallelisms are vastly different. Let's say if we want to use n number of processors for parallelism with distributed.gl, you have your code written and passed on to this so-called controlling process. And from this controlling process, n number of workers will be spawned, and that's where your computation will be distributed among. And the results from these distributed workers will be summarized back to the controlling process. If there's any I/O, that will be done on the controlling process as well. As for MPI, we know that your code is essentially executed in n number of ranks. Sort of the n identical copies of your code are running at the same time in parallel with communications in between all these ranks. Due to the very different structures of this parallelism, the way to run Julia on the HPC, especially on let's say small EM will be different. Here are the examples for your Slurm setup. With distributed, since it is a built-in Julia feature, all you need is to pass on the number of processors you want to use as a Julia argument. As for MPI, you really need to set up the srun command, and most of the time you also need to load the MPI module on the cluster. Now let's take a look at the Monte Carlo algorithm that I'm using for my research in physics. On the left, it's a very simple example of using Monte Carlo to estimate the value of pi with random numbers. Monte Carlo becomes way more powerful and efficient for higher dimensional problems. One of the higher dimensional problems in my daily research is the ultra cold atom physics. So the physics is when you cool the atoms down to a very, very low temperature, you can individually manipulate these atoms, and they put the atoms into this potential that looks like a washboard. From the picture, it looks like even simpler than the example on the left. It's just 5 balls in the 5 pockets. But however, the atoms, they follow the laws of quantum mechanics. Once a quantum mechanics is involved, things becomes more tricky. And to describe the system, you need a wave function, which is constructed as a superposition of all possible configurations with these atoms and a corresponding coefficient. This problem often solved as a matrix eigenvalue problem. When you have the Hamiltonian matrix here works on the wave function according to the Schrödinger equation, you can obtain the energy of the system. For example, this 5-atom system, the Hamiltonian matrix looks like this. It is very sparse. However, the problem of the scaling of this matrix gets worse very quickly. For example, if you have 200 atoms, it's still a very small atom, a small system in your real life. The dimension of the matrix can get to a very large number that's larger than 10 to the power of 100. It is a huge number. Say if you convert all the atoms in the entire universe and convert all the atoms into silicon and build a big memory, it still won't be enough to store this matrix. That's why we need help from this Monte Carlo algorithm. Instead of building the matrix, we're sending a set of workers to sample this matrix. And we have defined these configurations as D1, D2, D3, so on, and we let these workers move between these configurations to give you the information you want. So the typical number of workers we use is 10 to the power of 6 to 10 to the power of 9. Obviously, it's way smaller than 10 to the power of 100, but it's still a decently large number. We want to parallelize it and run the calculation on the HPC. Imagining these black boxes are the individual computer processors. It's really nothing stops us from parallelizing them with the obviously communication in between them. So now let's quickly talk about profiling. Say once you have your code written and you want to know how well it runs and how to potentially improve your code. And profiling is a thing you can do to help. In the background is actually a simple example of Julia code with native Julia profiler. On the right panel, you see some analysis of which line spends how much time in this code. It looks pretty straightforward, but however, our problem, our code is way more complex than that. The code is longer, and obviously once the parallelism is involved, it becomes harder to profile. Luckily, we got help from Massey consultancy team. After facing all these challenges, including that Julia is a very new language, we have limited guidance and experience that you can search from internet, and it is pretty new, not just for me as a coder, it also is kind of new for the Massey managers. So we really actually look at this thing together, and the outcome was pretty great. And we tried many things, and we found out eventually this Intel Reaching Profiler works fairly well for Julia with parallelism. And this Reaching Profiler is fairly easy to use as well. However, one thing is a special build of Julia from the source need to be built by the Massey managers to enable the reaching feature. For now, the Julia with version 1.2 and 1.4 have the reaching functionality enabled. Let's take a quick look of what kind of information you can get from this reaching. Here, I'm showing the CPU utilizations for both distributed and MPI. The first thing you can spot for the distributed.gl is if you count the number of processors, you have 3, and the spec we only ask for 12 CPUs. That's because, let's recall the paradigm of the parallelism. We always have this controlling process here that initialize the parallelization and finalize it as well. And for the majority of the time, you can see the other 12 CPUs are busy computing your stuff as shown as the brown regions in this band. And let's look at the MPIs. And firstly, if you count the number of processors, it's only 12. And seemingly, they are all busy running and computing stuff. Like you can see the brown region that took up all the space. But there is an extra yellow region you can see from the plot that actually shows you the MPI rank is waiting for the communication between all the ranks to be done. So there is a small inefficiency there. And this usually referred as the MPI barrier. So let me quickly summarize what we learned. So with distributed, it's actually good for relatively small scale computation and especially on the single node that doesn't involve the communication across multiple nodes. And it's good for serial I/O. And another advantage is it is a pure Julia code with no hardware dependency. You don't have to load any external packages. So it is great for cloud computing. And as we saw today, it will be good for reproducibility as well. As for MPI, it really utilized the fast infinite band interconnection on the cluster. and the communication across nodes is very efficient. Also, if you want to do parallel I/O, means say you want to write into a single file from many different CPUs at the same time, the MPI is good for it. Coding-wise, MPI requires some more low-level thinking. As Julia is designed for sort of easy and convenient coding, the MPI requires you to put a little bit more effort to think of how to actually code it efficiently. As the outcome from the profiling and the consultancy project, we managed to get our code optimized to a 20 times faster. It is we kind of switched to the MPI for our code because it is really efficient on the NZRSE clusters with its fast interconnection. Also, there are other factors involved in the optimization, like we learn more and new features of Julia, and we have written better code as well. If you're interested and want to check out the code, it's called remote.gl, available as open source on GitHub. If you want to learn more about the consultancy project, you can check out the link here. So with this quick hand-waving summary, Julia is a really modern and powerful language, and there's multiple parallelism really available for you to choose and based on the need. There's still many new things to explore, and we still have a lot of things to learn. And lastly, the NZRSE consultancy project has been very helpful for us in terms of improving our code. I'd like to acknowledge our founding body, the Marsden Fund from the Royal Society, and the help from the brand group and CDCP members from Massey, and also the NZRSE consultancy team. And lastly, thank you for your attention.
Well, thank you. Thank you, Mingrui. We got a question for you from Joachim Brandt. He says, would you recommend using Julia for such a project? I suppose the question is for you, Mingrui.
Yes, I would definitely recommend it. So it is so Julia has when the creator creates a Julia, it has the design for scientific computing. So it is definitely designed for this scientific project like that. Also, it is the parallelism is also as a initial part of the design, so it is quite optimized if you use a distributed feature, and multi-threading is also implemented there. Yeah. And the good thing is there are other also like external packages like data frames and like file IOs. They also can help you to improve the performance.
Yes, another interesting question is probably for both of you. Hillary asks, do you think Julia will eventually overtake Python for high-level scientific coding?
Oh, that's a tough question. I will say it very frankly, probably yes. Like for me personally, I have no intention and no reason to switch back to Python. And one feature of Julia I put into my slide is the just-in-time compiler. So that's really a feature which is also available in Python. And don't take me wrong, it is a built-in feature is Julia and pretty efficiently speed up your code. That means that instead of like Python and normal script and like you write your code and Python read your code and do your computation. So the Julia reads your entire script and compile into machine code and then execute it. So it is indeed faster with this just-in-time compiler.
What do you think, John?
Well, I'd say often languages coexist. Fortran isn't gone, put it that way. So, I mean, I wouldn't say you need a winner. It's coexistence is the winner.
Absolutely. It really depends
Fantastic.
on your need.
Wolfgang got a question. Yeah. Wolfgang got a question for you. It seems, do you think that Julia is easier to use and faster than, for example, Python, Cython, or Number?
I have
So probably the question for Mingrui or John or both of you.
Yeah, I have a relatively limited experience with Python coding. So at least for me, Julia isn't hard to learn, and obviously is pretty pleasant to use.
Yeah, I'd say the learning curve
Okay.
of any language depends on your background and your current familiarization. Okay, so again, I don't, it's kind of like saying, is there a better learning style, right? Every individual has a learning style that works for them. And I think it's kind of the same with learning programming languages as well. There will never be just one programming language, okay? And anybody who gets into programming at all in their lifetime is going to learn more than one. And I think it's unproductive to seek the one language. It's a moving, growing thing.
Yes, it is. It's a very interesting observation from both of you, Mingrui and John. It's fantastic. What questions do you have from the floor? We still have a few minutes. And Hillary, of course, appreciates both of you to say that as a good answer. Is there nothing else? No, there's not a question. Some final words from both of you before we wrap it up. We still have got a few minutes left.
Well,
perhaps John.
I'd like to again stress what I think is the importance of interdisciplinary collaboration, and especially for anyone who's in a position to make higher level academic administrative incentives for this kind of thing to happen. Because I think academic competition, whether it's within a discipline or between and all this, is counterproductive. Really nice things happen cross-discipline. I think that's exceptionally important.
Great, yep.
Also, it's fun. You know, as somebody who's specialized in computer science, for me, spending too much time in one narrow focus is boring. I like learning biology. I like learning mathematics. That's what keeps life exciting and engaging.
That's fantastic. Yes, certainly it is, isn't it? Yeah. What would you like to say, Mingrui?
I absolutely agree with John's point. And like me, I'm learning physics, but spending a lot of fun on coding as well. It's very enjoyable. Yeah.
Oh yeah.
And from my point, from my Julia programming's point, I like to encourage people to give it a go. Well, and at least it is free, so it's nothing stops you to try that out. And yeah, again, like coexistence of all languages is definitely great. So give Julia also a chance. Yeah. That's it.
Just one other thing, that there's a question from the floor, but just a quick question that the slides will be made available, I believe, at some point for both of you, if someone is interested to take a look at this after the events. And this is, of course, recorded. So those of you who have registered for this will be able to view them later. Great. So we're near the end of it. If there are no more questions, then I'd like to really thank both of you for a fantastic set of presentations. Greatly appreciate it. And I certainly enjoyed the session. So over to you.
Thank you.
You're welcome. Thanks for having me.
Yes, thanks for hosting.
Thank you. Thank you. Thank you for your fantastic presentation. Thank you very much. Grateful. Okay.